Take a look at that number on your screen. Black women are 40% more likely to die from breast cancer than white women, despite fewer cases being diagnosed. That's according to the American Cancer Society. The research being done to better understand this concerning statistic and what it will take to overcome the hurdles to reduce that number. This is Bench to Bedside.
From the research bench to the patient's bedside, this is Bench to Bedside with your host, Vice Chancellor and Director of the University of Kansas Cancer Center, Dr. Roy Jensen.
Good morning. I'm Dr. Roy Jensen, Vice Chancellor and Director of the University of Kansas Cancer Center. Joan Lewis-Wambi, Associate Professor in the Department of Cancer Biology here at the University of Kansas Medical Center, and Dr. Danny Welch, Associate Director for Education at the Cancer Center, are here to talk about breast cancer research. Also joining us is Tonia Yelder, who is a breast cancer survivor. Welcome, Tonia. How are you doing today?
Having me here on the Bench to Bedside program.
So, Tonia, you were diagnosed with something called triple-negative breast cancer. And we will ask Dr. Lewis-Wambi to break that down for us in a minute. But how did you find out that you had that particular type of breast cancer?
Okay, thank you for that. I performed a self-breast examination from a shower hanger given to me by a volunteer in my community. She told me, don't forget to check, and those words stayed with me. I performed a self-breast examination and found an unsmooth cluster of bumps underneath the skin. And after visiting with my OBGYN, I was sent to a specialist for a biopsy. The results confirmed I tested positive for triple-negative breast cancer, and it took some time to absorb the news.
I would imagine so. Dr. Lewis-Wambi, tell us more about Tonia's particular type of breast cancer, triple-negative. How is it different from other breast cancers?
So what we know about breast cancer is that it's a disease of types. And so there's the estrogen receptor positive breast cancer, which is the majority of breast cancer. About 70% of patients are diagnosed with the estrogen receptor positive. And so it's driven by estrogen. And then there's HER2 positive. It's another hormone receptor positive breast cancer. And then there's triple negative. Triple negative represents about 15% of cases. And what it means is that patients who are diagnosed with triple negative, their tumors do not express the estrogen receptor, progesterone receptor, or HER2 receptors. And these are the three classic receptors that we use in terms of determining what types of treatments that we would give to patients. So triple negative, even though the frequency is lower than the ER positive breast cancer, it's harder to treat. And so there's a lot of effort to try to understand what makes it so aggressive, what makes it so difficult to treat. So that is the type of breast cancer that Tonia has. So currently we don't have any targeted therapies that are available for triple negative because there's no known driver of that tumor.
I see. So a study last year found that black women are actually 3 times as likely to develop triple-negative breast cancer. Do we know why that is? Is it diet, biology, environment? Is there a specific thing that accounts for all of that?
I think that is the question that's really been appealing to me and so many others. It's why is there such a higher prevalence of triple negative? And there is a lot of evidence that diet plays an important part in that. So obesity is linked to triple negative. It also has to do with your environment. So a lot of studies are focused on the interaction between the environment, but also the biology of the tumor. So what is it within the tumor that is driving it to give it that aggressive phenotype? And that's what we're interested in the lab, is understanding really the biology of the tumor, because we know that triple negative is different in African American women compared to Caucasian women and other groups. So what is it about the cancer in African American women that makes it so aggressive and makes it difficult to treat? And really to understand the biology of the cancer cells, what is happening within the cancer cell. That will give us more clues as to what is going on and how that interacts with the environment.
Mm-hmm. So Dr. Welch, we know that there are major racial disparities in terms of cancer incidence and mortality. Do we understand what are the causes behind those disparities?
Well, the short answer is not really well. Mm-hmm. There have been a number of studies that have been linking socioeconomic issues, diet, access to medical care, the insurance, all of those things that Dr. Lewis-Wambi just mentioned. But increasingly, there are a number of papers and publications showing that there are actual inherent genetic differences that exist with people from different ancestries. So African American is different from African American, Hispanic, and there are different ethnic groups within the Hispanic populations. All have different genetic makeups. And if you control for living in the same neighborhoods, same rough diet, roughly the same economic stuff, there are still differences in the kinds of cancer that people develop, as well as the aggressiveness of those cancers. Mm-hmm. And where we, meaning the field at large, are still trying to identify what those genetic differences are so that they can be exploited.
Mm-hmm. So if you're just joining us, we're here with Dr. Danny Welch, Dr. Joan Lewis-Wambi, and Tonia Yelder. Remember to share this link with people you think might benefit from our discussion. Use the hashtag #BenchToBedside. So let's switch gears a little bit and talk about what is being done in the lab to better understand these disparities. And specifically, I want to turn to you, Dr. Lewis-Wambi. Tell us about your work with this disease.
So our lab here at KU Med Center is really interested in understanding the pathobiology of triple negative. As I mentioned before, we're interested in learning more about the tumor cells. What is going on within the tumor cells? What are the genetic components of that cell that allows it to be more aggressive than other subtypes of breast cancer? And then looking specifically at African American patients. So we've had the great fortune of being able to get clinical samples from the biosample repository here at KU Med from African American patients and from Caucasian patients, and really to compare those two groups to see what are some of the genes, what are some of the proteins that are being expressed. And there are similarities between the groups, but there are differences. And we're focusing on what is similar, but also what are the differences, and then are those differences the key to helping us understand why one group is at a higher incidence or are less likely to respond to treatment? Because we know that even though African American women are treated with chemotherapy, which is the standard of care, they don't always respond as well as other groups. And so the idea is that maybe there's something inherent within the tumor that makes those tumors more resistant to treatment. Mm-hmm. So really understanding what is going on within the cancer cells, what is driving it, what are those pathways, what are those genes or proteins that are being expressed in those cells that help them to work the way that they work, and how can we identify targets to potentially develop drugs that can be used for those patients. And so we've identified a pathway, the interferon signaling pathway, that we hope is key to understanding that. And what we've discovered is that interferon signaling, or type 1 interferon signaling, seems to be disproportionately upregulated in triple negative from African American patients. And we've identified a subset of genes that are regulated by interferons, and interferons are cytokines that are normally produced in the body, and it's usually stimulated by viral infection. And so they're there to really stimulate your immune system to mount a response to that viral infection. And what we're trying to see is, why is this pathway dysregulated in triple negative in African American women? And how can we use that information to identify a potential target? And so this is how I was able to get in touch with Tonia, because we were interested in looking at triple negative and looking at this pathway specifically within African American women, and then how could it potentially benefit patients. And so I reached out to Pivot and discussed the projects that we had going on in the lab, and explained to them what we were interested in doing. And they were kind enough to arrange a meeting with Tonia and other members of Pivot, and I was able to explain the types of research that we're doing, the gene that we identified, and our efforts to try to target this gene. And so that has been our focus, really, to understand the biology of the tumor, what is going on within the tumor, and is there a way for us to identify a driver? Because that is the challenge with triple negative. It is a disease that's highly heterogeneous. There's no one driver of that tumor. Whereas we talk about estrogen receptor positive tumors, we know that they're driven by ER, or the estrogen receptor, or HER2 positive tumors, they're driven by HER2. And then there are drugs that are available that target those specific pathways and those receptors. For triple negative, we do not have a clear driver. There are many genes that are dysregulated in triple negative, which makes it very challenging to treat. So the idea that we've identified a gene, IFIDM1, that seems to be exclusively expressed in triple negative from African American women gives us hope that it could be a potential target. So we're focused on really understanding the biology of the tumor, and then trying to develop targeted therapies for that.
I see. So Dr. Welch, black women, it's also known, are much more likely to be diagnosed with metastatic breast cancer, which means when cancer is spread to other parts of the body. And I know that metastasis is certainly one of your great areas of expertise. Could you tell us a little bit about your research around the phenomenon of metastasis in cancer?
Sure. As you were mentioning, the major cause of death or loss of quality of life for any cancer is due to metastasis, or the spread to other sites. The triple negative breast cancers tend to metastasize very widely, and they will occur in a lot of the vital organs. So we've been focusing in my lab primarily on the genetics that underlie metastasis, mostly focusing on the genes that suppress the metastatic phenotype. And so for people who are listening in, basically these metastasis suppressing genes are kind of like the brakes in a car, and the brake system has been ripped out of the cancer cells, so they're basically able to just run and do what they do. Recently, we've been focusing some of our attention on a different set of genes inside of a cell. So everybody's aware, I think, of the Human Genome Project, which is the 3 billion bases of DNA that's our genetic makeup. But all of our cells in our bodies actually have a second genome. It's an organelle called the mitochondria, and that's where a lot of our food gets turned into energy that people can use for whatever the cells use. And it turns out that when one looks at mitochondrial genes, it's a very small genome, but there are specific spots within that genome that can be used to define ancestry. So several years ago, a fellow named Doug Wallace identified that there was a mitochondrial Eve, because we inherit mitochondria only from our mothers. And as humans migrated out of Africa into Europe and Asia and so on, there were changes that occurred. And we can look at those specific changes in mitochondria, and those can define ancestry, which we can colloquially colloquially call race. And we've made mouse models that mimic this and show that certain strains of mice prevent metastasis for multiple kinds of cancers. Other mitochondria from certain strains of mice promote metastasis. And we're hoping to be able to translate that to better understand the disparities for people developing metastatic cancer. Mm-hmm. The other reason I think your question was asking about, if you were asking about why black women will die more often. And that comes back to that disparities issue that I raised before. Besides the inherent differences that different ancestries or races have, there are also differences in access to care, diagnosis, and quite frankly, for years, people haven't gotten all the information to the various ethnic groups and racial groups. And that is something I know we're working on heavily. You as the director know that better than anybody in the Cancer Center. And if it's okay, Dr. Lewis-Wambi mentioned something called Pivot. Mm-hmm. And I just think for people who are listening in maybe for the first time, that's a program at the KU Cancer Center. It's called Patient and Investigator Voices Organizing Together. And it's a recognition by the researchers and physicians at KU that it's important to have patients providing input so that our research and our clinical care are focused on what the patient needs most, not what's inherently just interesting to us in the lab. Mm-hmm. So,
Dr. Welch, obviously the mouse work that you described takes place in the lab.
Yes.
And but the ultimate goal, obviously, is to move those findings from the lab to patients in the form of a clinical trial. And I'd address this question to both of you. Why is it so important that we diversify the patients that we enroll on our clinical trials? Why is that such a critical issue?
I'll take a stab at that and then ask Joan to sort of chime in. Yeah. There are a number of differences that occur between the different races. And I'll just cite one. It's not about treatment per se, but in a way it is. And it was actually pioneered and discovered here at KU. And that is people trying to quit smoking. There's a drug that's been used, but it was much less effective in African Americans than it is in Caucasians. Mm-hmm. Certain chemotherapies, a lot of the drugs that are being used have to be activated in our bodies in order to become effective. And if there's a deficiency in an enzyme or an overactivity of an enzyme that activates those drugs, that can make a patient get sicker because of the chemo or get better. And knowing those things are critical. The other aspect relates to something that Dr. Lewis-Wambi brought up, and that is collecting specimens. So clinical trials, every patient gets standard of care, but during that time, they're also participating in a human experiment of sorts, not to experiment on the people, but so that every patient's information can be used to help patients in the future. Mm-hmm. And so every patient who's on these clinical trials is donating some of their cancer and some of their normal tissue so that enough information can be gathered in order to help patients down the road. Mm-hmm. So and in order to do that most effectively, we have to know how different groups respond. Mm-hmm.
So Dr.
I will absolutely echo what Dr. Welch said. I think it's very important that patients understand that they are represented. I think if they understand that, I know somebody else who has the disease that I have, and they are actually looking at what works for that person, they're more inclined to say, okay, they have my interests at heart. And I think that is important for us. Representation is important. Every group wants to feel as though they are being represented because they may have issues or concerns that are unique to that patient population that others may not fully understand. And so those issues can be brought forward and can be addressed in an effective manner. I have 4 daughters. Triple negative is something that I think about a lot. My mom, my sister, I have nieces, aunts. I have a lot of people I know who have been diagnosed with triple negative or other types of breast cancer. I've met lots of patient advocates, lots of survivors, several of them African Americans. And I love seeing that. I love to see patient advocates who are invested and engaged and are aware of what's going on, because that is the only way we can disseminate information. That's the only way we can make them aware of what is going on in terms of research. I think it's great that Tonia is involved with Pivot and involved with the Cancer Center. She has a voice. I think her voice needs to be heard. I think her working with me and other members in the Cancer Center helps us. It helps us to remember our why we're doing what we're doing, and to remain patient-focused in our research. I think as scientists, sometimes we get lost in the lab doing things that we think are super important to us, but I think we have to remember why we're doing the research that we're doing, that ultimately it is to benefit patients like Tonia and others like her. And so I think we have to invest in doing clinical trials that have a wide range of representation in those trials, because that's the way we'll get the maximum information. And that's the way we can make the most impact in terms of patient care.
Yep. So the project that you talked about earlier, would it even be possible if we didn't have diversity in terms of our patient population and those willing to participate in the
Oh, it would not be, because the differences that I saw were between the African American patients and Caucasian patients. The pathway we were able to identify was uniquely upregulated in the African American triple negative samples that we looked at. So it is important for us as researchers, if we want to do the types of research that we know is needed to be able to make that impact, we have to have access to clinical samples. We have to have access to tumor samples or serum, whatever it is that we need to be able to do those experiments. And we're not going to be able to have access to it if patients are not willing to donate their samples. And they're not going to be willing to donate if they don't feel vested in the research that's being conducted, or they don't trust the research that is going on. And so I applaud Tonia, because I think you're wonderful. I think what you're doing is great, and I think it helps other survivors. It motivates them, and it helps them to see that there are others going through this the same way I'm going through this, and it helps them to be more receptive to us when we come out and we say, okay, we found these interesting things. Well, if we have advocates who can say that we've engaged them in our research, they're more willing to listen to what we're saying. So it's a great system that we have here, having the patients involved in our research, because I think it really helps inform our decisions and help inform the types of research that we're doing, and hopefully it will bring about a positive change that we want to see.
Yeah.
So.
Can I just add one quick thing? I think
Sure.
just to emphasize something which was just said, it's a dialogue between the patient and the researcher and the physicians that are involved. It's all 3 groups have to be communicating all the time. And the Pivot program is an early step in that. It still can be improved, but Tonia's been a part of that, and I think a lot of the other patients have contributed to a lot of the different kinds of research.
So Tonia, now you took part in a clinical trial a while ago. Could you tell us a little bit about the process that you went through to help you decide whether or not to participate in a clinical trial?
Okay. When I went to see my oncologist for the very first time, and for my chemotherapy treatment, and we talked about it. And he told me that I had almost a 15% chance of a reoccurrence, and I was like, that's not enough. That's not good enough. So please figure something else out. So he left, and he came back about 30, 40 minutes later with a clinical trial study nurse. And then after they talked to me, they said, okay, all I heard was, now you have less than a 2% chance of a reoccurrence. And I was like, yes. So that's when I learned about clinical trial studies. And that was motivation enough to reduce the chance of a reoccurrence for me to have breast cancer a second time. I couldn't do it again.
I see. So now you have obviously continued to participate in the research process by joining Pivot, and we've mentioned that a number of times now. And Pivot is our patient research advocacy network. Could you tell us a little bit about your specific involvement in Pivot and how you've partnered with researchers in the Cancer Center, including Dr. Lewis-Wambi?
Definitely. So I have worked with rapid reactor teams. I've worked with research scientists. I've gone to the lab. I have been I've looked over lay abstracts, and I've written letters of support for different cancers, for esophageal cancer and breast cancers. And it's a community engagement where they care about my cancer experience. And so I give that information freely so I can help the next person or help someone else. And there are lots of different things that Pivot is involved in helping with the research. And I think that's just a wonderful thing. It's just a great organization to be a part of. And I hope that more people consider Pivot and try to service either caregivers or cancer survivors can play a part in the Pivot process.
Great. So, Dr. Lewis-Wambi, now we're going to turn to your side of the Pivot equation. And how did Tonia's perspective specifically impact your research and approach to the problem that you're trying to look at?
So, interacting with Tonia gave me, I guess, a new appreciation for why we're doing what we're doing. And we collaborated on a V Foundation grant. And the goal of that grant, that was a collaboration between myself and another faculty member here at the Met Center. And we wanted to have Tonia and other members of Pivot be a part of that grant. It's a grant that was focused on triple-negative breast cancer and early detection. And we thought that Tonia's experience would be relevant to our project because we're trying to target individuals who are affected by triple negative. And this was a health disparities grant. So it helped us to have someone like Tonia who has triple negative, who is African American. And she was very much interested in this blood test that we were thinking of developing using interferon signaling as an early marker for detection of triple negative and how it could be beneficial to patients, not only like herself, but others along that line. So I think partnering with her and partnering with Pivot helps me to understand why we're really doing this. What is the impact it's going to have? And it's not just doing research just to do research. It's the purpose of the research. What is the impact that research would have on patient care? And so I think for the longest time, I thought, we're doing all this great work in the lab. But since I've started serving on the Department of Defense Scientific Committee, and we have a lot of patient advocates on there, and it makes you see why you're doing what you're doing. And having patient advocates sit next to you who currently have metastatic disease, and they're so engaged in the research, knowing that it may not necessarily benefit them, but someone who will come after them. and they are well informed. They know about the research that's ongoing, and so it helps us to realize that what we're doing makes a difference. And so I would say to Tonia, thank you for being engaged, for being informed, for being willing to help us do the research that we're doing, because it would not be possible without patients like Tonia and others like her. So we need to have that open dialogue. We need to have that interaction, that collaboration, because that's the only way we can move research forward. And so that has really helped me. It's given me a different appreciation for why we're doing the types of research that we're doing and how impactful it can be.
Mm-hmm. So we're approaching the end of today's episode, and I want to ask one more question of each of you, and I'm going to start with you, Dr. Lewis-Wambi. What do you hope to accomplish with this project as we move forward in over the next 10 years?
I think really it's twofold. Understanding what is it within, and I know a lot of labs are studying triple negative. It's an area that everyone is interested in it, and we've made some breakthroughs, we've made some positive strides, but we haven't figured it out as yet. So my hope and my goal is to see if I can help to contribute to a better understanding of the disease. And in terms of that, how does the pathway that I've identified, the interferon signaling pathway, how does it actually contribute to TNBC or triple negative breast cancer aggressiveness? And why is it uniquely dysregulated in African American patients? And how does that impact not just the tumor, but the tumor microenvironment, so the immune system, because the immune system is actually very important in this whole process. And so the idea that the tumor cells are making interferon, which normally they're not supposed to, so why is it making interferon, and how does that impact the other cells in the surrounding area? And then how can we use that information to develop an actual target, and then design a drug that can target our protein of interest? Mm-hmm. Because ultimately our goal is to move our findings from the lab to the clinic so we can benefit patients.
Absolutely. Dr. Welch, where do you see your research going over the next few years?
Well, I think it boils down to two major goals, and they're related. We want to identify what exactly are those differences that exist between people of different races, and use that information to give people the most effective therapy that will benefit them the most, and not expose people to therapies that are going to be ineffective or that will make their quality of life poor. Mm-hmm. And that the information we're gaining can be applied in both those directions. But I would venture to say we're not quite yet ready to know exactly how that's going to happen.
Sure.
But that's the goal.
Yeah. Right. Well, Tonia, and last questions for you. But first of all, I want to thank you for sharing, your cancer experience, and it's so important to our audience to hear your voice. So my question is, what advice do you have for those who are facing a similar diagnosis, namely triple negative breast cancer?
Cancer does not discriminate. It impacts all races, all nationalities, all ages. I was 32 years old with small children when I was initially diagnosed. Self-breast exam saved my life. I encourage women to get mammograms and to perform those self-breast exams. If you are not old enough for a mammogram, please take better care of yourself. The BRCA gene that I carry, it was discovered later because I couldn't afford it. So insurance can only help with so much. But I do carry that BRCA gene that was discussed by Dr. Welch. But so perform those self-breast exams on your own once a month on the day of your birthday. If you're over 40, get them the mammograms every year. And there are clinics that will help people that don't have insurance. There's mobile vans that will come to the community and out there and help people. My church has done that also. Continue with clinical trial studies, and maybe the first one doesn't work, but don't give up and stay encouraged. Eat a healthy diet and take time to take care of yourself so you can live a longer, healthier life.
It's great advice. And Tonia, thank you so much for being part of this week's episode of Bench to Bedside. To learn more about genetic testing, visit kucancercenter.org/breast cancer. That's it for today. Join us next week at 10 a.m. for Bench to Bedside. Thanks for watching. Subscribe to our Bench to Bedside podcast, now everywhere podcasts are available.